Good morning, students. Hope you people are in a good state of health. Today, we'll take up chapter 8, How do organisms reproduce? And the topic that we will cover today is asexual reproduction. Now about the learning objectives of the session: reproduction, importance, types of reproduction, difference between asexual and sexual reproduction, different types of asexual reproduction. That means after completion of the session, you will be able to understand all these topics in a better way. Now let's start with the definition of reproduction. It is a biological process by which all organisms multiply in number and increase their population. In simple words, we can say that giving birth to like ones is called as reproduction. Now, the question will come in our mind why reproduction is necessary, why it is an important feature of living organisms. Reproduction is very necessary to maintain the existence of a species. It maintains the continuity of life. So, reproduction is necessary if we want to continue our existence as well as to maintain the continuity of life. Now about the types of reproduction. There are two types of reproduction: sexual and asexual reproduction. Asexual reproduction is a method of reproduction that involves only one parental organism. A single organism reproduces two or multiple organisms on its own. This is seen in all unicellular organisms, some multicellular organisms, and few plants. Now about the sexual reproduction. The mode of reproduction that involves two parental individuals, one male and one female. They produce gametes or sex cells which fuse to form a new organism. You must have read in class 8 that the gametes, male and female gametes, fuse to form zygote. Zygote multiplies to form a new organism. Now, the difference between sexual and asexual reproduction. It is a very important topic for board exam. You need to learn this table. Let's start with the differences. In sexual reproduction, it takes place in almost all types of organisms that are unicellular, including the higher plants, humans, and animals. While asexual reproduction occurs only in lower invertebrates, chordates, and plants with simple organizations. The second point of difference is sexual reproduction involves the fertilization process, and in asexual reproduction, fertilization doesn't occur. In sexual reproduction, gamete formation, that is called as gametogenesis. Genesis means formation. So gametogenesis means formation of the gametes. Gamete formation occurs in sexual reproduction, while in asexual reproduction, gametogenesis does not happen. In sexual reproduction, two parents are needed, while in asexual reproduction, single parent forms the new organisms. In sexual reproduction, germ cells proceed to perform sexual reproduction, while in asexual reproduction, somatic cells are the normal body cells, proceed to perform asexual reproduction. So this is some common differences among sexual and asexual reproduction types. We may include one more point that in sexual reproduction, because there is recombinations of genes, so variations occur. While in asexual reproduction, there is no genetic recombination and so no variations. Now about the types of asexual reproduction. The main important types are fission. It is of two types: binary fission and multiple fission. Regeneration, budding, vegetative propagation. Tissue culture includes in vegetative propagation, and spore formation. Let's start with the discussion on these important types of asexual reproduction. First, we'll start with the fission. Fission is the division of parental body. If the parent body divide itself and form new organism, this process is termed as fission. Generally, the lower category organism or unicellular organism shows the process of fission to increase the number of the organism of their population. It is of two types: binary fission and multiple fission. Bi means two. That means in binary fission, one parental cell divides to form two daughter cells. As you are able to see in the given image, that there is one parental amoeba and the parental cell constrict in between, and the constriction deepens down, which divides the parental cell into two. The cytoplasm as well as the genetic material or the nucleus simultaneously constricts, and finally the constriction reaches the middle point and the two daughter cells get separated from the single parental cell. This is called as binary fission. Generally, the unicellular organism like amoeba and paramecium are supposed as immortal in nature, and the reason is that because there is no loss of protoplasm and they divides their complete protoplasm or complete content into the daughter cells. So because of the continuity of the complete protoplasm, they are considered as immortal in nature. Now about the second type of fission, that is termed as the multiple fission. In multiple fission, many daughter cells are formed inside the mother cell or the parental cell, and finally the parental cell bursts to release the daughter cells. Because many cells are formed, so it is termed as multiple fission. Now about the major differences between the binary fission and the multiple fission. In binary fission, the division of cytoplasm and the division of nucleus takes place simultaneously, while in multiple fission, first the nucleus divide and finally the cytoplasm divides to form many daughter cells. In binary fission, there is no cyst or cover formation, while in multiple fission, cyst formation takes place. Cyst is the hard cover like structure surrounding the parental cell. So, In binary fission, only two daughter cells are formed from one parental cell, and in multiple fission, more than two daughter cells are formed from one parent. So these are some common differences between the binary fission and the multiple fission. Now about the second type of asexual reproduction, that is termed as regeneration. It occurs in lower category of organism. Here the organism is planaria, which is a type of flatworm. When by anyhow the planaria and its body divides into different parts, each part has the ability to form the complete organism. This ability or this process is termed as regeneration. Also, there is a process similar to it termed as fragmentation, which is seen in plants. The algae and the lower category of plants show regeneration when by anyhow, because of some accidental purpose or because of the pressure of water and whatever, if the alga or the filament divides into different parts, each part has the ability to form a new organism or new algal filament. So that process is termed as fragmentation, which is similar to the regeneration in the animals. Now about budding. In budding, the parental body forms an outgrowth of protoplasm. Finally, the different parts develop in it, and the small bud, it cut off from the parental body, start a free existence, increases in size and become an adult organism. So, budding is the process which is commonly seen in the coelantrates like hydra as well as in yeast. Here, beneath the tentacles, anywhere over the body, a small outgrowth appears. Later on, different tentacles and mouth appears on that small bud, and finally the bud cleave off from the parental body or get separated from the parental body and become a new organism. Now about vegetative propagation. It is a type of reproduction in which, except flower, any part of the plant is able to form a new plant, like a stem, root, leaf, etc. Here we have taken the example of bryophyllum. In the leaf, there are notches at the edge, and each notch has bud. When the leaf fall off on the soil, in contact with the soil, the buds germinate to form new plants. So from a single leaf we get many plants, and so there is rapid multiplication in the number of the organism of the population. So this type of reproduction in which any part of the plant except the flower arises the new organism is termed as vegetative propagation, and the part involved is termed as vegetative propagule. Here the leaf is acting as a vegetative propagule. Similarly, in the potato plant, the potato tuber that we consume as a vegetable is having eyes or buds, and when you keep the potato tuber in dark for some days, then the green types of shoot start arising from the eyes, and it shows that the potato tuber is able to form new organisms. So potato tuber is the vegetative propagule in that process of vegetative propagation. Now about the tissue culture, which is a part of vegetative propagation. Here what happens is that in tissue culture, which is also termed as micropropagation, any cell tissue of the plant is able to form millions of plant with similar feature to the parental plant. If I want to get the exact features of a parental plant in the new organism, and I want to get it in a very short duration of time, and the copies are around millions of copies, then we can proceed for tissue culture or micropropagation. Here, if there is a plant which is desired in all features, we take out a tissue or cell from it, and that part is termed as X plant. EXPLANT is the spelling. Now the X plant is transferred into a nutritive solution which has all the essential nutrients required for plant growth, and we have added into it a plant hormone, that is auxin. Auxin is the root promoting hormone. When we keep the X plant in the contact of nutritive solution, it rapidly multiplies. The huge mass of cells, where the cells are undifferentiated, all cells are similar in nature, that huge mass of cell is termed as callus. CALLUS. That callus later on, under the effect of plant hormone auxin, start forming roots in each cell. So we get multiple cells with roots. And finally we transfer the section or the sample into another nutritive solution which is having the plant hormone cytokinin, and cytokinin is a shoot promoting hormone. And finally each cell with root forms shoot, and we got millions of small plantlets. We transfer these plantlets to the nursery beds, and within a short duration of time, from a single cell or from a tissue, we get multiple copies which are exactly similar to the parental copy. We even can get disease-free plant from a diseased parental plant. If we take out the apical meristem from the plant and use it as an X plant, then we can easily get disease-free plants from a diseased parental plant. So the image is also showing how in the nutritive solution we get a plantlet from the tissue. Now the spore formation, it is mostly seen in the fungus. Here the spores germinate on the nutritive medium and forms filament-like structures. The single filament is termed as hyphae, and when it multiplies to form a mass of filaments, that is termed as mycelium. It is found in the fungus. Now, in favorable conditions, some hyphae start going in the vertical direction towards the upper side, and as shown in the image, towards the upper side, and they bear a small ball-like structure which is termed as sporangium. Inside the sporangium, the division takes place and many spores are formed. Later on, the sporangium bursts to release the spores. Because single parent is involved, so it is a type of asexual reproduction, and the liberated spores again settle on the nutritive mediums, either on the open food sources, and finally they germinate there to form a new fungus. So We have discussed the definition of reproduction, types of reproduction, that is sexual and asexual, major differences between sexual and asexual reproduction, and the important types of asexual reproduction. Hope you have attained the learning objectives for this session. Take care of yourself.